Morning students. So we continue with this topic, excretory products and their elimination. So yesterday we had discussed some of the important excretory products found in the animal kingdom, the various excretory organs, and of course we moved on to the human excretory system. So I had given you the structure of the nephron. Now let us take the various parts of it, the histology of the nephron. Okay. So moving on with histology of nephron. So first part, as we have seen, was Bowman's capsule, right? Now Bowman's capsule is a cup shaped structure. So just like any cup, it will have an outer lining and an inner lining, and that is important because this has appeared in the questions also, you see. So outer lining is made up of simple squamous epithelium. Outer lining is simple squamous. The inner lining, this is also simple squamous, but these are specialized cells, and this inner lining is made up of podocytes. It's made up of podocytes. Now podocytes, you see, these are cells which are giving off number of processes which are further branched out. This is how the cell is there, and these processes you can see just like we put our fingers together, that is, it's called interdigitation. So these are interdigitating, and these are surrounding the capillaries, you see. So the glomerular capillaries, we'll see, they are surrounding. So this podocyte, this is one podocyte, you see, and this is a foot process. This is a foot process and foot process. These interdigitate, that is just like fingers, they are going in, interdigitate, and the gaps which are in between, those are called slit pores, forming slit pores. Okay, so this is about these one. Let me see, it's clear. Yes, the board. Yes, it's not. I hope, please let me know if the board is hazy or anything, so I can just adjust it. I hope it is clear and you are getting the sound and everything. Okay. So now this is one, that is the Bowman's capsule. Now with the Bowman's capsule, as we had seen that within the Bowman's capsule there is glomerulus. So let me mention it here itself. Although we have mentioned yesterday that nephron is having two parts, that is the renal tubule and the glomerulus. And glomerulus is within this Bowman's capsule. So let us mention it here itself, right? So here coming on to glomerulus. So glomerulus is a tuft of capillaries, which you have seen. They are formed by the branching of afferent arteriole, and these capillaries, they join to come out of the Bowman's capsule as efferent arteriole. Okay, so this is tuft of capillaries, and these capillaries are quite different from the rest of the capillaries of the body because they have fenestra. Okay, so these are highly permeable due to fenestra. Now what are fenestra? Let us see that. So you see, in any capillary, if you will talk about, let us say this is the capillary here, and in this capillary the inner lining, here this is the basement membrane, the inner lining is endothelium, and endothelium is simple squamous epithelium, you know that, and squamous epithelium is arranged like floor tiles. There is hardly any intercellular space left out, but here the arrangement is not like this. Rather, the cells are arranged and there are spaces in between. So the cells are not arranged end to end, rather there are spaces here, and as a result, you see, now any substance which is from in the blood has to move out. That means it has to cross only the basement membrane. It doesn't have to cross the cell boundary. So if one barrier is reduced, so you can very well understand that it is going to be, the permeability is going to increase. And this capillary, you see, it is surrounded by podocytes on the outside. So the foot processes of the podocytes, these are surrounding them. We are just making a cut section diagram so that you can get an idea. So this is how the arrangement would be. So in this gap here that you see, this gap between the cells, this is what is called fenestra. Fenestra, which literally means window. So more are the windows, more will be the passage possible, right? So fenestra, these are present in this glomerulus. And then here, as you see, this thick one, this will be the basement membrane. And this is the foot process of podocyte. Foot process of podocyte. And between this space here, we have just now mentioned, it is called the filtration slit or the slit pore. So just like in respiration, as we had mentioned, the alveolar epithelium, the basement substance plus the endothelium together, that is making the respiratory membrane or the diffusion membrane. Same way here, these three things together, they form the filtration membrane. Okay, so similarly here we can use the term filtration membrane. Okay, filtration membrane. So filtration membrane would be the glomerular capillary endothelium. Okay, so endothelium plus basement substance or basement membrane, I am just writing short BM for it, and here the podocytes covering around it. Okay, so they are slit pore. So any substance which has to pass out of the blood has to cross these three. So then that would be the filtration. So that is a super sieve, you can say, that is formed. Now coming on to the next part, that is PCT, the proximal convoluted tubule. This you have once again done in the chapter in animal tissues. So PCT, this is lined by brush bordered cuboidal epithelium. Brush bordered cuboidal epithelium, and each of this cuboidal cell is having many microvilli, and within the cell there are many mitochondria also. You can understand, because here lot of absorption is going to be active absorption, so that's why mitochondria are needed for the energy supply. Okay, so many mitochondria and many microvilli and mitochondria present. Okay, then coming to Henle's loop. Now Henle's loop, whether it is descending limb or ascending limb, the histology is same. But yesterday you must have seen I had drawn two parts. One was thin segment and another one was thick segment. Now the thin segment is lined by squamous epithelium, and the thick segment is lined by cuboidal, of course simple epithelium. These all are simple epithelia. Okay, cuboidal epithelium. So just in short we are going into it. Then coming to DCT. Now DCT is same as PCT, same type of lining, but the microvilli and mitochondria are fewer. Okay, so same as PCT but has fewer microvilli and mitochondria. Okay, that is there. And then coming on to the collecting. Now you see the part of the collecting duct, we labeled it, but the part which is lying in the cortex, that is called the collecting tubule, and then when it comes into the medulla, that is the collecting duct. Okay, so collecting tubule and the collecting duct. Tubule and duct. So this is once again simple cuboidal epithelium, and this one, as I told you, collecting duct is not taken as a part of the nephron. Fine. So this is the basic histology. Then we have two types of nephrons. Okay, so there are two types of nephrons. One is the cortical ones, and the other is juxtamedullary. juxtamedullary. Now these are more, these are 85% of the total nephrons and these are 15% of the total nephrons. So the basic difference, I'll just draw it here, you see. Suppose this is the cortical part, this is cortex, and here this is the medulla. Okay. So the cortical nephron is smaller because the loop of Henle is short. So its major part, you see, is lying in the cortex. It is not that it is not going into the medulla, but very little part will be going into the medulla. While juxtamedullary, juxta means junction. So it starts at the junction of cortex and medulla. It starts here and it has much longer loop of Henle, which is going deeper into the medulla. So that is one basic difference. So it's very much there in the diagram, we needn't write it. Then another important difference is here vasa recta. Usually the books write that it is absent, but NCERT has mentioned that it is poorly developed. Let it be. Vasa recta is poorly developed. Okay. And here vasa recta is well developed. Vasa recta well developed. Then another thing we can add on is that cortical nephrons, you see, they are responsible because kidneys are not only the they don't only have the function of excretion but also maintaining osmoregulation. Now osmoregulation is maintaining the amount of water and the various electrolytes, that is ions, in our blood plasma. So there has to be a proper concentration, not only in the blood, overall extracellular fluid, because there would be some tissue fluid also, and blood and lymph are there. So overall body fluids, this amount of water and ions should be in a proper balance within a certain range. So the kidneys have to ensure that how much urine is produced. In that urine, otherwise lot of water and salts, if they are lost, then there is a problematic situation, right? And if there are too much of it, then also it's a problem. So it maintains it, or it is helpful in maintaining the blood volume under normal conditions. So maintains blood volume in normal conditions, and this one maintains blood volume in under stress. Under stress, and stress for the kidney is, stress for the body here, this is not mental stress, not psychological stress we are talking about, it is water scarcity. Like especially when in summers, you see, you haven't got water to drink for a long time and you have been sweating, losing lot of water already. So in that case, the urine needs to be concentrated so that more loss is prevented, right? So under stress, that is water scarcity. Okay, so this is one. Then before I start with the urine formation, let me take one more thing that is associated here, although that we'll be discussing later with the hormonal regulation, that is about the juxtaglomerular apparatus. Okay, so I think by this time people have logged in and people are awake and they are taking down the notes. Okay, so next thing. Now you see here yesterday when we had drawn, this was how the Bowman's capsule is. I am not drawing the whole part, and you have seen that the descending layer means the Henle's loop goes down, and when it comes up, before it means the descending the DCT starts, it is very near to this place. Okay, so let us just mention this is the DCT. This is of course the Bowman's capsule. Okay, and here from one side, I'll just make it a little smaller. Okay, so this is the Bowman's capsule here. So one side you see it is the afferent arteriole coming in. This afferent arteriole is then branching into number of capillaries, right, which is the glomerulus, and when it goes out from here, it is a narrower blood vessel, which is the efferent. Okay, and here in the DCT, here at this point there are certain cells here. Okay, we'll just mark a very simple, this is like a spot. So let us mark this thing. This is afferent arteriole, which is broader, as you can see. The efferent arteriole, which is narrow. This is the ascending limb of Henle's loop. Now these all short forms you see when your NCERT is writing. If you will see, read the summary in the NCERT, you will find Henle's loop, they have written as HL. So I think every these short forms, at least which the NCERT gives, you should be knowing about them. So this is the DCT here. So DCT just at this junction at the glomerulus, here it is having certain cells, which, and that group of cells is called macula densa. Macula densa. Now macula densa, they are very much cuboid, these cells only, cuboidal cells, but that would be a spot, or macula means a spot. So it is just this part of the, suppose you are standing inside the duct, so in this duct, which is cylinder, but on that cylinder you can imagine a spot there, the spot which is towards the glomerulus, that is macula densa. And here on the wall of the afferent arteriole, here there are some special cells, okay, here on this only, which are called JG cells. JG cells, that is juxtaglomerular cells. Okay, and this entire structure, this, that is, we'll just mention, this is called the juxtaglomerular apparatus. So we can mention this thing. Juxtaglomerular apparatus, or in short just JGA. Okay, so juxta, that is once again formed at the junction of glomerulus, Now what all is present in it? One is juxtaglomerular cells, JG cells. Now JG cells are present, as you can see in the diagram, they are present on the wall of the afferent arteriole. Okay, so these are present on wall of afferent arteriole, right? And these cells, they secrete renin, and mind the spelling of renin, this is R-E-N-I-N, not with a double N here, that was an enzyme in the stomach. Okay, renin, and another hormone is there, that is erythropoietin. Erythropoietin, the other day we mentioned, for RBC synthesis. Okay, so erythropoietin stimulates the bone marrow cells to make RBCs, and renin, we'll see, has a role in regulation of body fluids, right? So that is one, JG cells. Then another structure which is contributing to this juxtaglomerular apparatus, that is called macula densa. So macula densa, these are sensors. You see, these cells are sensors. Location, you can very well see it is on the DCT. And what they do, because you see from here the glomerular filtrate is passing. And in this glomerular filtrate, they will be sensing the level of NaCl. Okay, because NaCl, once again in the blood you have seen that NaCl is the most important salt over there, the principal cation and anion. All our muscle contraction, nerve impulse conduction, that has important, Na has important role. So that's why Na has to be maintained in proper amount. So here if too much of Na is reaching, so that means the initial part of the nephron, that is the PCT, the ascending limb, they have not been able to absorb Na, and all that Na is now going to be lost in the urine. So that's why immediate the process will start to conserve it. Okay, so these are sensors to glomerular filtrate, okay, so they will sense it, and these stimulate JG cells to release renin, Okay, this is one. And there are few cells here in between. There is nothing vacuum like in our body. There won't be any vacuum. There are some cells here in this gap here. those are called lacy cell, and till date exact function is not known. But why I am telling you here, because this could be all of these kind question, that juxtaglomerular apparatus includes, and you have 1 JG cells, 2 macula densa, 3rd lacy cell, and of course your answer will be 4th, that is all of these, or more than one option is correct, like that kind. Okay, so I'll just mention this, lacy cells, and these are unknown function as yet, but these are also there. Okay, so this was also one part of the anatomy. Now with this we are through with the anatomy part, and now we can start with the process of urine formation. Okay, so next thing, now before I take up urine formation, just one more thing, that is about osmolarity, because in this we will be mentioning osmolarity once and again and again. So osmolarity, what we mean by osmolarity? So osmolarity is same what you study in chemistry as molarity, the same thing. That is the amount of solutes, in simple words if we put it, amount of solutes dissolved per liter of solution. Okay, so this is amount of solutes dissolved per liter of solution. And since you know our body fluid is a very dilute solution, in the blood you have seen 90 to 92% is water, and the electrolytes in it, they are hardly 0.9%, the ions. So here the osmolarity is, we measure it in a unit that's called milliosmol per liter. So normal blood osmolarity, is 300 milliosmol per liter. Okay, and this we'll just write in this way, MOSM, O capital here and liter. This is how it is written. Okay, so kidneys, when we are talking about urine formation, they won't only be removing the nitrogenous waste of the body, but they will also be maintaining normal blood osmolarity. That's what is osmoregulation, right? So the first step in the urine formation you see is ultrafiltration. So next, coming to this urine formation, starting with ultrafiltration. Now ultrafiltration, you see, it's a totally a passive process. No energy is used here. So just like you use sieve at home to filter maybe tea or anything, so it's a passive process and it is non-selective. Non-selective, that means anything which can pass through the filtration membrane, if its size is so much that it can cross the filtration membrane, it will cross. Right? It is not selective. If it was selective, then the next two steps were not needed also. Right? So it's a passive, non-selective process. Okay? And so what is guiding it? The driving force is the differences in the pressures. Okay? So passive non-selective process driven by glomerular hydrostatic pressure. This is the main pressure which is responsible for the filtration to occur. Glomerular hydrostatic pressure. Okay? Now this GHP, in short it is called GHP. GHP develops due to this thing, the arrangement, the anatomy, that is the broader afferent arteriole and narrower efferent. So you see, from a broad, if the vessel is broad, that means more blood volume is coming through it, and this large amount of volume of blood now has to squeeze through thinner blood vessel, so certainly a pressure would be generated. So there is a bottleneck created, and since these glomerular capillaries are so permeable, the fluid will come out from here and come into the Bowman's capsule. So GHP, this is due to broader afferent arteriole and thinner efferent arteriole. Broader afferent arteriole and thinner efferent arteriole. And if this anatomy is reversed, just an assumption, GHP will not develop, because from a narrow thing to move on to a broader one, it's easy, right? So pressure will be not generated. And this pressure is of the order of 60 mm of mercury. Now this is the pressure for filtration, but there are certain pressures which are opposing filtration. So let us mention pressures that oppose filtration. That oppose filtration. Now these pressures, one is BCOP, that is blood colloidal osmotic pressure. Blood colloidal osmotic pressure. Now you see the colloids which are present in the blood, they are holding back water, they are not allowing that water to go out. And those colloids are what? Proteins, the plasma proteins, and in the plasma proteins the most abundant proteins are albumins. And this also once has been a question. The blood colloidal osmotic pressure is due to, so it is due to plasma albumins. Okay, so this is due to plasma albumins. Okay, so you will, in the blood there are so many proteins, but the maximum amount of protein are albumins, and they are contributing to this blood colloidal osmotic pressure. Now this pressure is of the order of 30 mm of mercury. Okay, then there is another one, that is CHP. CHP, this is capsular hydrostatic pressure. Capsular hydrostatic pressure. Now this pressure develops due to, because you see this cavity here between the outer and inner wall of the Bowman's capsule, here also some fluid will be there. As I told you, there is never any vacuum, there is some fluid here, and that fluid will not allow more fluid to come into it until unless it has moved ahead into the PCT, right? So that is the capsular hydrostatic pressure, you see. So this is due to fluid already present, due to fluid in Bowman's capsule, right? And this pressure is of the order of 20 mm of mercury. Okay, so you see this is the pressure which is favoring filtration, but there are two pressures which are opposing filtration. So the effective or the net filtration pressure would be the difference between the two. Okay, so I think I can clear this side now. I have explained everything. So now we can have this. The net or the effective filtration pressure. Okay, and this is, you can write NFP or EFP, any such short form we can write for it. So this will be GHP minus BCOP plus CHP. So the pressures favoring filtration minus the pressures opposing filtration, and you see this is 60 and these are 30 plus 20, that is 50. So your value would be 10 mm of mercury. Now once there was a question in AIPMT many years back, there the question was the net filtration pressure is, and out of the various choices there wasn't 10 mm was not there, the lowest choice was 25. So in that case, of course, use this 1400 cc here, that the lowest one which was given, you have to just answer for that, right? Because this is for humans we are studying, but other animals, all animals don't have the same pressure, so they never mentioned it is human, because in some of the old books where human, instead of human physiology, rabbit or frog and so many other physiologies they were studied, so the values are different, of course. So it was kind of that question. So you just have to use, you shouldn't be totally perturbed that the value is not there which I have studied. you see out of that it is a very, very low value. So out of the different values given, the lowest one was to be given, since the answer none of these was not there. And mostly you can say 99.9% of the times in the NEET exam, you won't have a choice of none of these. So you have to select your answer out of the 4. So at that moment you have to see out of the 4 which is most appropriate. Okay. Now coming to this, this is filtered, fine. Now what gets filtered is called the glomerular filtrate. Okay. So glomerular filtrate. Now what does this glomerular filtrate contain? I'll give you a very simple thing. You can just mention this is plasma, the blood plasma, because the blood cells are not being filtered. So whatever is there in our plasma, it gets filtered except the proteins. So it is plasma minus the proteins. Okay, so that means this filtration membrane just now we talked about, it will allow water, electrolytes, glucose, amino acids. Even in the blood there are some water soluble vitamin C and all those are there, urea, uric acid, all that is getting filtered. So instead of making such a long list, we can simply write it is everything that is present in the plasma except the proteins, that is in the glomerular filtrate. And how much filtrate is being formed, that would be per unit time, that is GFR. So GFR is 125 ml per minute. Okay, so this is the glomerular filtrate normally. Then another is how much blood the kidneys were getting. Okay, so blood supply to kidneys, and when I say kidneys, that means to both kidneys together. So blood supply to the kidneys, you will see it is 1100 to 1200 ml per minute. So your kidneys actually get more blood supply than your brain. The brain gets 750 ml per minute, kidneys get this supply. Okay, and out of this, what is actually getting filtered is the plasma. So out of this, the renal plasma flow, if we see, renal plasma flow, that is amount of plasma that flows through the kidneys per minute. So you know that plasma is, we have done that in the blood, plasma is 55% of the total blood. So if you take 55% of it, you will get 650 to about 700, this is just a round of ml per minute. So this is the amount of plasma, this is total blood. Okay, so from that total blood, if we just see how much plasma is passing, and out of this plasma 125 is getting filtered. So that will give us filtration fraction. Filtration fraction. So filtration fraction would be GFR upon RPF, and you can put into 100, so you'll get a, instead of fraction you'll get a percentage. So you can do it 125 upon 650, you can do with 125 upon 700. So you will get a range around 0, or you can say 16 to 20%. Now NCERT hasn't given this range, they have just they have mentioned that as much plasma flows through the kidneys. So if you take 20%, so 20% would be 1/5. So out of that 1/5 gets filtered. So as much of the plasma flows through the kidneys, that 1/5 is getting filtered, right? So and the blood supply also, you see the cardiac output is 5 liters per minute, and here almost 1 liter per minute is going. So kidney are getting the supply of 1/5 of the cardiac output, even that way you can take. So this is one. Then next thing, after this filter, now since in the plasma there are number of things which the body needs, they are not to be excreted out. So now comes the role of tubular absorption. Tubular absorption. So I'll just you see, I'll give you a simple form, that is, suppose this is the entire renal tubule, just simply we are making it, and this is the blood goes here, goes into the glomerulus, then here it is coming out. Okay, and from here the glomerular filtrate which has formed, that is passing through the nephric tubule. Now from this glomerular filtrate, some few things are absorbed back into the peritubular capillaries. The capillaries which are surrounding the renal tubule, those are called the peritubular capillaries. Yesterday we have marked it in the diagram. So few things will be absorbed back into the blood, that is returned to the blood, that's what is tubular reabsorption. And few things are going to be secreted. The lining of the nephron, you see here also the cells are producing certain waste, and they can extract waste from the blood also, so they will be secreting few things into the filtrate. So you see the distance, this thing. So this is reabsorption, something which is going back to the blood, that is reabsorption, and one which is taken from the blood and put into the filtrate so that it can pass out of the body, that is the tubular secretion. And you can very well understand both these things are going side by side simultaneously. It is only for our study that we take them one by one, both of them. So I prefer to take them together, and in each part of the renal tubule we will see that what is being absorbed and what is being secreted. So first take down everything what is there in the board, clear so that I can clear the entire board, but then I can use it properly. Okay, so take it down. By this time, I think this side must be over. Right? Are we done here? Okay, I've left some part. So now you see one column we can have the region of the renal tubule. Another we'll take what is getting reabsorbed here. So tubular reabsorption, then tubular secretion, and lastly of course we'll see that what is the osmolarity of the glomerular filtrate. That is it isotonic to our blood, is it hypertonic, that means more concentrated, or it is hypotonic, that is less concentrated. So that can be a narrow column there. So I think by this time everybody is through with this. Okay. So you see, starting with PCT. PCT, maximum reabsorption you see is occurring here itself. Almost 65% of the glomerular filtrate gets reabsorbed. Okay, so 65% of glomerular filtrate, just I am writing GF, it gets reabsorbed. Now in this, what all is getting absorbed? You see, 2/3 water, or you can say 70%. Right, 2/3 is 70%. 70% of the water gets absorbed, 70% Na, 75% K. Now here one more thing. In the NCERT it is mentioned that potassium is secreted. Small amount is secreted, but mainly it is reabsorbed here, right? So you will see the diagram where from the nephric tubule the arrows which are going out, arrow which is going out, that means this substance is getting reabsorbed because it is coming out from the tubule, and it, of course it will be going into the bloodstream, right? And the arrow which is shown going into this, that is for secretion, that is for secretion. So in the diagram you will see the diagram is correct, but in the text portion they haven't written it, so don't get confused there, no problem. Okay, but of course if you do get exact statement, since it is from the NCERT, we'll have to take it as correct. but there is absorption also. So this is there. Then apart from this, there is chloride. I'm just not writing the percentages, just for the important ions and water I'm writing. This is there. Then 100%, this I can certainly mention, 100% glucose, amino acids, okay, even vitamin C, all this is getting reabsorbed here. And once again, the absorption part is very similar that I had taken up with you in the digestion. That is, in digestion we had seen glucose was absorbed by secondary active transport. That means along with Na, it goes in, and that Na will then be sent out of the cell by expenditure of energy, putting out 3 Na into the blood and 2K from there into the cell, right? The same process. So active and passive, you can see very well there. Tubular secretion, as far as it is mentioned, because you see even the cells are producing certain waste. So ammonia is there, hydrogen is there. Even if we have taken any medicines, so you see these medicines are foreign chemicals for the body, and those are also excreted out. So all those drugs, any toxins which have been present in the body, all this will be secreted from this area. And osmolarity, because you see both things are happening. And when you say 2/3 water, and some students, they do get confused that 70-75% K is there, but 70% of what? Total ionic concentration in the plasma is 0.9%. So 70% of that 0.9%. So it is very less. So you will say that here it is isotonic. The osmolarity of the glomerular filtrate is isotonic to the blood plasma. Okay. Then next thing, coming to Henle's loop. Now Henle's loop, first part, that is descending limb. Now descending limb is permeable to water only. So from here only water is absorbed, nothing else. Water is absorbed, and water absorption once again would be by osmosis. No secretion here. Now suppose you have a solution with you and you evaporate water out of it. You remove all that water. So what happens? Your solution becomes concentrated. So that's why you will see in the NCERT diagram also that as we are going down into the medulla, they have written certain numbers, 400, 600, 800, 1000, and at the tip of the hairpin bend there is 1200. So as the glomerular filtrate is passing down into the medulla, water is coming out and the concentration of the filtrate keeps on increasing. So that means it is becoming hypertonic. Okay, here the osmolarity is going hypertonic. Then coming to ascending limb. Now ascending limb is permeable only to ions. It is present ions. So here sodium, potassium, chloride, calcium, magnesium, so on. All these are being absorbed. And secretion we can of course mention for H ions, that is there. And so whenever I write H ion, I'll have to mention bicarbonate, because this is a pair. Bicarbonate is absorbed and H ion is released, because the product, this is once again, remember, respiration, same way. So even in the cells of the nephric tubule, carbon dioxide is being produced, which is forming H2CO3 combining with water, which splits into or dissociates into H plus and HCO3 minus. H ions are excreted out because too much of H ions will make the body fluids acidic, while bicarbonates are absorbed. Okay, so these go hand in hand. And now only the ions are going out, water is not being moved. So what happens to the solution? The glomerular filtrate becomes hypotonic. And let me mention here, because if glomerular filtrate osmolarity we are mentioning, okay, not the blood osmolarity. So here, but this is of course with respect to water, with respect to blood, the osmolarity is hypotonic. So that means less than 300 it will reach. Then coming to DCT. In DCT also you see this one, this goes hand in hand. Here, but rest of the absorption you see is facultative, or you can say conditional absorption. That will depend on the presence of the hormones. Okay, so there is conditional absorption conditional absorption of water, that we'll just be mentioning because of ADH. There is potassium ions, these are there, sodium ions, chloride ions, these all are there, and there is here there is secretion of potassium, absorption of Na, right, conditional absorption. Even in this area there is absorption of calcium, you see, that will be because of PTH. Okay, so we can mention this, all the hormones which are acting over here. ADH acts here, aldosterone acts here, aldosterone is for sodium absorption, and also PTH acts here, which is not of course mentioned in your NCERT, but I am giving you. So this absorbs and the secretion is of this thing. Okay, I think this will be yes. So but still in the cortical regions you will find this is once again isotonic. And lastly, coming on to the collecting duct. Here also water does get absorbed, that is conditional absorption, so it's almost similar to DCT in most of the sense. But one more thing is there. In the medullary part, the part of the collecting duct, of course, that is in the medulla, it is permeable to urea also. So from here urea is also absorbed. So urea from medullary part. Urea from the medullary part. I hope it is visible. Yes. And as far as secretion is mentioned, H ions, K ions. So depending on the hormones, if the hormones are present, then lot of water is going to be absorbed here, otherwise not. So accordingly we have the ability to produce dilute urine as well as concentrated urine. And here for the first time now the glomerular filtrate is called urine. So once it reaches collecting duct, so we can mention this glomerular filtrate is now termed urine. And this we have because we are after all osmoregulators, we can change the osmolarity of the urine so that our blood osmolarity remains constant. Because you see, if you drink lots of water and you are not sweating also, you are sitting under the AC, so what happens? More dilute urine is produced. While when you go out, you are sweating a lot, the urine becomes concentrated. So that's what osmoregulation is, and we are osmoregulators. We maintain a constant blood osmolarity irrespective of any kind of place we are. And how are we doing it? We can change the osmolarity of the urine so that urine can be concentrated or dilute, so that blood osmolarity or blood volume is maintained, right? So here it can be hypo or hypertonic. And maximum hypertonic it can reach is 1200 milliosmol per liter, and you will see that is at the deep end of the medulla, that is the level that reaches 1200, and our blood osmolarity I just now mentioned with you is 300. So that means we can concentrate the urine 4 times. As much our blood osmolarity, we can concentrate it up to 4 times. So I think this will suffice. This is everything is over here, and you can take it down. And as far as I told you, absorptions are concerned, water is always absorbed by osmosis, so it's a passive method. Sodium and potassium, wherever you see here, sodium is absorbed, potassium is secreted. The process is same, expenditure of 1 ATP, 3 Na out, 2 K in, the same process, and that is of course active transport. That is active transport. Hydrogen, for hydrogen also there are special proton channels that also takes energy, so that is also moved with energy, right? So glucose, secondary active transport, sodium dependent transport it is. So the processes are exactly the same as we have done in digestion. Okay, so I think you can finish it off, and then we can talk a little about urine. Right? And sometimes if you do get terms like conditional absorption, so conditional absorption is, you can also use the term facultative, and here the absorption doesn't depend on any hormone. This much absorption is going to take place of water and other things. So this is obligate absorption. It doesn't require a condition. If conditions are there, it can go beyond 70% of water, but 70% is going to absorb, right? Now, by the time you are noting down, I can pose a question to you and let me see some people answer. In the presence of ADH, in the presence of antidiuretic hormone, maximum water is absorbed in. Okay, I think you have heard the question properly. In the presence of ADH, maximum water is absorbed in, and your options are PCT, descending limb, DCT, collecting duct. Okay, come on, answer. Okay, there is one. Yes. I am waiting for your answer. By the time you note down, you can answer it also. Itu has answered PCT. Yes. So by the time I am just clearing the board, waiting for your answer. I want some more people to answer. Okay. Let me see anybody else is answering or not. PCT, Ekta, Shraddha, Himani. Right, very good. You see, we have already mentioned that 70% water is absorbed in the PCT. So that means rest of the parts of the nephron are absorbing out of the remaining 30%. So the number means the amount cannot be beyond that, right? So PCT, this is just a question to confuse you, that in the presence of ADH or even if it is in the absence of ADH, your answer is going to be the same. Maximum water absorption occurs in PCT. And similar question in a different way, it had appeared in the NEET, I think few years back, 2-3 years back, that if we remove the PCT, what happens to the urine? So if you remove the PCT, the rest of the nephron cannot absorb that 70% water, right? So the urine is going to become very dilute. All that 70% water will pass out in the urine. So it will be highly dilute urine. Okay, so now coming to this urine, an adult in a day produces 1 to 1.5 liters of urine, which normally has a pH of 6, although that pH can vary according to the diet and all, but average pH is this. And this one, 25 to 30 gram of urea is eliminated in the day. Okay, and what does it contain? So constituents, first let us mention normal constituents. Normal constituents. So normal constituents, of course it is water, huge amount is water, 95%, and in remaining 5% there are a number of things, that is urea, uric acid, creatinine. The major is urea, of course, that's why I have written like that. Creatinine, hippuric acid, these ions that we had just now mentioned, potassium ions, H ions, some sulfates, phosphates. These all are there, and so many other compounds also, but basically this. And even you see some of the hormones. That's why you see through the urine test we can check for the presence of a particular hormone as well. So this is just the major things I am mentioning. Then if we talk about abnormal constituents, so it could be glucose, that is in diabetes mellitus in this disorder, and the condition will be called glycosuria. So you see any term ending in urea, that means the substance is present in urine. Okay, glycosuria. Then similarly ketones, if these are present. So this is once again diabetes mellitus, I'm just writing DM. So this is in DM, as well as long-term starvation. And long-term starvation, the condition of course is called ketonuria. Because you see in diabetics there is a lot of glucose in the blood, but the body is not able to metabolize that. So it seems as if their body cells are starved and they are using fats. So by the breakdown of fats, ketones are produced. And then another one we can mention is albumins being present. That is the first sign of kidney damage. Albumins. So this is, you see, condition would be glomerulonephritis, that is inflammation in the glomerulus and the nephron, and this is condition is called albuminuria. And when there is more damage, even larger proteins can start appearing, so that would be simply proteinuria. Then another one we can mention is hematuria. So hematuria is blood in urine, or you can say RBCs, because blood, when we are talking about it, is RBCs are coming out, RBCs being the most abundant formed elements. And this can happen in case of injury, if there has been injury to the kidney, or that injury could be internal, not an external injury, somebody hasn't beaten that person, but rather it is renal calculi. So renal calculi, you see, these are kidney stones of calcium oxalates. So these kidney stones, when they pass through the ureters, so they damage the lining, causing blood to come out, right, from the minor calyx, major calyx, they are damaging over there. So because of that this happens. So these are just a few of the abnormal constituents that we have talked about. There is a huge list, so that's why you see number of times when there is some kind of ailment, the doctors, they prescribe a urine test, and through that urine test also the pathological state can be detected. Now next, coming on to next part, that is one thing that is left out, concentrating the urine. So for that you needn't go into detail, because for MCQ based paper the body has the mechanism to concentrate urine by counter current mechanism. Okay, so the urine is concentrated by counter current mechanism. Now this counter current, the term itself means when there are when there are fluid flowing in any duct or a tube in opposite directions. So when it flows in opposite directions, there is always the gradient difference will also always be there. That means diffusion can be an ongoing process, because you see if two fluids are moving in the same direction, there is diffusion. Suppose there is a substance which is more in one of the tube, so it will by diffusion move to the next one, but that will be possible only till equilibrium is established. So once equilibrium is established, further diffusion will stop. But counter current is when they are in opposing directions, and as a result from one side the fluid is moving away, the other side fluid is being added, the diffusion gradient is always present. So this counter current is formed in our nephron by two things. which contribute. One is of course the Henle's loop. So Henle's loop, you have seen that descending limb going into the medulla and the ascending limb coming up. So the glomerular filtrate is moving in opposing directions, so that is Henle's loop, and across this Henle's loop it is the vasa recta. The vasa recta. These two structures are responsible for concentrating the countercurrent mechanism they are forming. And how is it being formed? Because you see, for absorbing water there has to be a gradient. Water is moving passively, undoubtedly. It is moving into the medulla, and from the medulla it will go into the blood, the peritubular capillaries. But in the medulla there should be something so that water moves in. And so in the medullary interstitium, now let me just mention it in short here. So you suppose this is the descending limb, this is the ascending limb. So here fluid is going down, here it is coming up, and surrounding it the vasa recta is there. Here the blood flows down, here blood is flowing up and then moving on into the venule. Now NCERT has taken this and put it here, taken it out, but this is how the arrangement would be. So here you see once again countercurrents are made. So there are multiple countercurrents, countercurrent between the descending and ascending limb, and countercurrent between the vasa recta and the Henle's loop. And this space here, this is not empty space, here there is interstitium. So this interstitial tissue has lot of NaCl and urea here, and it is because of NaCl and urea collected over here that water will be moving out passively from the descending limb. So this NaCl and urea needs to be accumulated here, and that is done by that this ascending limb is transporting NaCl actively. So it is because passive transport would have been up to certain extent only, but active transport can be against the gradient. So lot of NaCl it is transporting, which is filling up over here, and we have just now mentioned that in the collecting duct the urea passes out passively. This urea also comes here, from here to here, that also gets collected, and this way the urine is concentrated. So in this way we can just mention that in the interstitium what is contributing for the absorption of water, so that is NaCl and urea. This is the only question that you can get from it. So needn't go into much detail. The structures responsible and the substance responsible. Purpose is of course to concentrate the urine. So that will do. Then coming on to the other hormonal regulation for this osmoregulation. Okay. So next thing, that is osmoregulation. Now osmoregulation is, as I told you, that is regulation of water and electrolytes in the blood. Okay. Now suppose a organism is in certain medium, like suppose let us say this is fresh water, and in this fresh water there is an organism. Now fresh water in comparison to the cytoplasm is dilute. So this, sorry, I am going the reverse. This is inside, it is hypertonic because the concentration in the cytoplasm comparatively is more, and fresh water of course this will be comparatively hypotonic. So where will the water move? Water will move into the cell. So water is moving into the cell, so such organism will have to keep on excreting water. Lot of dilute urine it will have to produce to maintain it. And the other condition, if we see, suppose this is marine water, and in the marine water if the organism is there, now marine water has lot of salt in it, so here this is hypertonic. In comparison the body fluid is hypotonic. So what happens? Where will the water move? The water will move out of the cell. Okay, so the cell is going to shrink. So both habitats have a problem, and we are living on land. In the land surrounding it is air. Air has little amount of water vapor, humidity is there, but still in comparison to the body fluids it is very less. So once again we are also losing water. We are losing water, so we have to retain that water. Now in marine habitats, some animals, what they do, they maintain their body fluid concentration same as the marine water. So here the blood and the sea water, they are isotonic, so they don't have any problem. And if such a thing happens, those are called osmoconformers. Conform means to change according to the situation. They don't have a stand, their own stand they are not having. So their body fluid concentration is changing, it changes according to the external medium, so that there is no problem to them. That is osmoconformation. And osmoregulation would be that we have to change the urine concentration so that our body fluid concentration remains same. Okay, so anyway, I'll talk about osmoregulation in us. So regulation of water and electrolytes in the blood, and in this case let us take the few situations, like if blood volume decreases, so along with that we can write so many things. NaCl in the blood is less, NaCl concentration decreases, and with this we can relate blood pressure decreases. And if all these are in the decreased state, you can also relate it that GFR is also going to decrease. So even the GFR is decreased. Okay, so I've put all the conditions together. Now in such conditions what happens, this is sensed by our JGA, and in that case the JG cells are stimulated to release renin. So now in this condition, all these conditions I have combined. So in this, JG cells release renin. Now renin, what it does, it acts on one particular protein, angiotensinogen. Now you once again see it's ending in ogen, so it is inactive. So this is present in our blood always. It's a plasma protein, and if it is a plasma protein, its source is liver, because all the plasma proteins except the antibodies, they are synthesized and secreted by liver. So angiotensinogen, this is plasma protein from liver. Now this protein is acted over by renin, so that it converts into, just like in enzymes you had seen, pepsinogen to pepsin, so on. So that's how it converts to angiotensin, but it is angiotensin 1 as yet. Angiotensin 1 is intermediate, it is not the active form. The active form is angiotensin 2. In short, we write angiotensin as ANG, angiotensin 2, and this is by an enzyme ACE. So let me put what is ACE. ACE is angiotensin converting enzyme. And this enzyme is produced in the lungs. It is in the lungs interstitium. So you see, when the blood flows through the lungs, so this all is inside the blood, of course, it is inside the blood vessel. So there angiotensin 1 will get converted to angiotensin 2. So this is in lung interstitium. Okay. Now this is the active form. Okay, so you see, this is here the activation is in 2 steps. So this is the inactive form, this is the intermediate form, and that intermediate is finally converted to the active form, that is angiotensin 2. Now this angiotensin 2 has major actions. Okay, what it does, 2 important things at least I'll mention. So angiotensin 2, number 1, it stimulates the adrenal cortex for release of aldosterone hormone. So this one stimulates adrenal cortex to release aldosterone. I'll just be writing what it does. And secondly, angiotensin 2 also acts on the PCT and it increases sodium absorption from there. Now you see, sodium absorption is already occurring there in the PCT. We have already mentioned 70%, but it will go beyond that. Okay, so this one stimulates PCT for increased absorption. That's why I'll just writing this way so that there's no confusion. So for increased absorption of Na, and Na along with the Cl it is absorbed, so NaCl also we can write. And when sodium is absorbed, you know there is a gradient created outside, so water will flow out by osmosis. So Na and of course water also. So this is major thing that we can talk about angiotensin 2. Yes, one more function I would like to write, that is this angiotensin 2. You see the name, angio word comes for blood vessel, tensin constricting, so it's a vasoconstrictor also. So this vasoconstrictor, so by vasoconstriction you see what is happening, that the blood pressure will rise. Blood pressure will rise because of vasoconstriction, and GFR which was decreased, this GFR is going to increase, but it is not going to increase beyond 125. It is just going to increase to the normal level. So that is also one thing which is important. So this way it raises blood pressure and GFR rises to normal. So I am writing that so that there is no confusion. Okay. Now let us talk about aldosterone. Now aldosterone you see is a steroidal hormone. So this was a protein here. Now we are talking about a steroid. So this is a steroidal hormone. Steroid hormone. In simple words you can just mention it is sodium retaining hormone. This is what is said about it, sodium retaining hormone. So where does it act? The target is DCT, collecting tubule and the duct. Okay, so this one stimulates DCT, collecting tubule and collecting duct, so that there is reabsorption of sodium, and with sodium there is passive absorption of water as well. And one more thing is happening because of it, there is secretion of K. So that completes the role. Okay, so this is what aldosterone does, and these three things combined, this is what is called RAS, that is renin angiotensin aldosterone system. So this is like a concert, one after the other they are acting. Okay, so this is the RAS system. And RAS system, when is it activated? I have given you all the conditions which you can get in the exam. Okay, so all of them combined. So any word they give you, you will be able to answer. Are we clear with it? Okay. Now along with this or apart, it can act along with or even separately. Another hormone is there, that is ADH. So once you are through with this part, I can just remove it. Okay. So next thing, let us mention ADH. So ADH is antidiuretic hormone, or also called vasopressin. Antidiuretic hormone or vasopressin. Okay. Now this ADH, it is secreted by hypothalamus, but it will come into the, from the nerve endings, it will come into the pituitary, from where it is going to be released into the blood. So released by posterior pituitary. Now next question is, when is this ADH released? So this ADH, you see, it is released. So this is secreted or released when the condition is when blood osmolarity when there is too much of water. You see what will happen to the blood osmolarity? The blood osmolarity will be more than 300, right? So when blood osmolarity rises, that is, it is 300 plus, okay, and so in this case, you see, this is sensed by the hypothalamus. So ADH is released. What it does? ADH, yes, when it is 300 plus, that means water is less, and there are more of solutes, so that means the water has to be conserved. So this one helps in conserving water, by concentrating the urine, So you see the DCT and the collecting tubule and the duct are naturally, they are impermeable to water. Okay, so DCT, collecting tubule and duct are impermeable to water, otherwise are impermeable to water, but due to ADH, these become permeable. So from here now water can be absorbed. Okay, so water will be absorbed, and as a result, as soon as the blood osmolarity comes down a little less than 300, 295 or so, further ADH release is going to stop. So when the blood osmolarity is high, it is going to be secreted, and like you see, you take lot of water, you drink lot of water, so what is going to happen to your blood osmolarity? Now there is more water in the solution, so osmolarity is going to decrease. So ADH secretion is also decreased. And one more thing, you see here also the name vasopressin. So ADH is also a vasoconstrictor. Okay, and lack of ADH causes a disorder, that is diabetes insipidus. Now this is different from diabetes mellitus, where the problem is because of excess glucose in the body, insulin problem. Now here the urine is in large volumes of urine are produced, but they are not having any sugar in it. Okay, so large volume of frequent urination, but urine is without sugar, is without glucose. Okay, that is there. Then now these two systems, they are maintaining our blood volume, and they are very much under negative feedback. They are controlled on there as soon as the system is able to come back to normal, but still water and NaCl is so important for the body that there is one alternative also, that if the system fails, there is another thing to take care of. So that is ANF, atrial natriuretic factor. Now this one, this is a hormone secreted from atrial walls of the heart. Okay, and when it is secreted, when blood volume is more. If the blood volume is more, there is increase in NaCl, increase in the blood pressure has increased. In this condition, it is secreted. So what does this ANF does? ANF is a vasodilator. So you see, it is antagonistic to the RAS and ADH. It's a vasodilator, so vasodilation will reduce BP. Okay, so it decreases blood pressure. Then it opposes RAS as well as ADH. And since it is a vasodilator, now here you see GFR is increasing, but this increases beyond normal, above normal, more than 125 ml per minute. So this one raises GFR above normal, and so more urine is produced, and more urine, you see, natriuretic. Natrium is the Greek name of sodium. So natrium passing out in urine as well as more water in the urine. Okay, so more Na and H2O passed out in urine. Right? So this is how our body regulates these amounts. Okay. Then another topic that is passing out of the urine, you see, that is micturition. That is a reflex. Let us just mention that quickly. So next one, coming on to micturition. So micturition is voiding of urine. Okay. So if we see the urinary bladder, so in the urinary bladder, this is how it goes, and within it, inside, if you are seeing, there is going to be a triangular structure here, which is called a trigone, and here there are oblique openings of the ureters coming over here. Okay, like this, and here there are two sphincters. Okay, so let us label this much part at least. So you see, this part is the dome shaped. In many organs we use the name fundus. This is body, and here it is the neck, and this is the inner one, you see, is involuntary sphincter, urethral sphincter. This one is voluntary sphincter. Okay, this is of course the urethra. Now here the wall of the urinary bladder is having inner layer, you know, transitional epithelium is there. Outer to that there is a muscular layer in which both longitudinal and circular muscles are present, and we can just mention detrusor. Those are called detrusor muscle. Detrusor muscle in muscularis. Why it is called detrusor muscle? Because they are so arranged that when they contract, they will be pushing down the urine. So they are arranged in such a way that it is coming into this trigone area. It is totally compressed, and the urine will go out. And the openings here, you see, these are oblique openings to the ureter. They are not having any valve, but since they are oblique opening, they get compressed and urine cannot flow back into the ureter. Okay, so this is one thing. Then apart from that, in the wall of the urinary bladder, there are certain receptors. These are called the stretch receptors. Okay, there are stretch receptors in the wall. So I'll just mention it here. Stretch receptor in wall. So when the bladder is full, the bladder has a capacity of 500 ml, and it can hold, because it can stretch, it can hold even up till 800 ml, but in that case it becomes painful. So okay, so stretch receptors are stimulated when bladder fills. Stretch receptors stimulated when bladder fills, and this impulse from here, it is going through parasympathetic fibers. Parasympathetic fibers, they carry this message to, from here, these parasympathetic fibers, these are going to the micturition center. So this goes to Micturition center. Now this Micturition center, normally the involuntary centers, you know, they are in the medulla, but some of the lower body centers, they are in the spinal cord. So this one is in the sacral region of a spinal cord. So Micturition center in sacral region of spinal cord. So this gets this message. So from here, what is going to happen is, I'll just rub this side. This one contracts detrusor muscles, sends commands for contraction of detrusor muscle, contracts detrusor muscle and relaxes the involuntary sphincter, the inner sphincter, relaxes the inner sphincter. Now these two things, you see, they are under the control of parasympathetic nervous system, but the external sphincter, which is the voluntary one is controlled from the cerebrum. For that the command will be coming from cerebrum, and that's why you see the micturition is it is called a reflex undoubtedly, but it is a learned reflex, because when newborn babies, as soon as their bladder fills, they'll just pass out the urine, they'll void the urine. But by the time the child reaches 2 years of age and they are literally trained for it, and from between 2 to 3 years they will learn to control their bladder, because if no washroom is available, the person of course has to hold the urine. So that becomes a learned reflex. So here the relaxation of the external sphincter or the voluntary sphincter is from the cerebrum. So relaxation of external sphincter is from cerebrum, and now of course the urine is voided. So once there was a question in this NEET exam that if all these stretch receptors are removed, then what happens? Now in that case, of course, the micturition reflex cannot work, because everything starts from here. If there is no stretch receptor, the micturition center will not get a message that bladder is full. It won't be there. So bladder keeps on filling, and where there was one option, there is normal filling of the bladder, that cannot also be there because it will be overfilling. And in that case, micturition won't be there, but because of the pressure of the urine, the sphincters are not able to keep closed, and they'll start relaxing and urine will start trickling out. So the person will have no control over the urine, and this sometimes, you see, it does happen if people have been in an accident and the nerve has been cut. There has been a spinal injury, the nerves which are carrying this message to the micturition center, that has been cut. So they lose control over their urination. So they are not able to do that. Okay, so that is a condition that arises. So that kind of question has also appeared. From this chapter, there are a number of, you see, thought questions. So you have to be very, the concepts have to be clear. And we are ending with the time. But anyway, disorders, more or less, I have mentioned. Some of the accessory structures which are also involved with excretion. Okay, so other excretory organs, if you see, like it is mentioned for lungs. So lungs are exhaling out carbon dioxide, which is at the rate of 200 ml per minute, and along with that we do lose some water vapor also, which of course is a loss for our body, but it does happen. Okay, then there is liver. So liver, we have seen the bile pigments being produced. Bile pigments, then even sterols are there, cholesterol, that is, and then breakdown products of the steroid hormones degraded here. So all that is also lost along with the bile juice, it is removed. Then similarly the sebaceous glands, through that waxes, some hydrocarbons, these all are there. Sweat glands, Sweat glands, of course, the main purpose is not excretion at all, rather it is temperature regulation, but along with that some wastes are also removed. So there is major is water, 99%, more than 99% is water, but there is some NaCl also, some lactic acid also, so on, number of products are there. And even the NCERT has mentioned through the saliva. You see, when you take medicines, the taste in the mouth goes so bad, because saliva after all is from the cells, and if something has reached into the cell, they will be producing it in their secretion also. So some nitrogenous waste, even some urea, it does come out in the saliva also. But that is not the major thing once and for all. The major function of the saliva is once again in digestion. So I'll stop here for the day. The dialysis part that we are left out with, that is the one major disorder, renal failure, which is, and how it is treated. So that we'll do in the next class and move on to the that is locomotion and movement chapter, right? So we can put a stop here. Thank you.